All right, let's go ahead and get started. Hello everyone, and thank you so much for joining our workshop today on cell differentiation. We have Sreethu Sankar here with us, who is a product manager at Proteintech, and he'll be offering some insights on how the quality of your growth factors can impact the integrity of your differentiations. But before I pass it off to Sreethu, I did want to just share a few company slides to introduce Proteintech to you. So, Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass many different research areas. And we also offer immunoassays such as ELISA kits and IHC kits. We also offer cell separation kits and antibody labeling kits as well. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors, which is what Shritha will be talking more about today. And what makes our humankind growth factors and cytokines different is that we express all our proteins in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, which then ultimately affects their bioactivity. We also have our Chromatech branded nanobody-based reagents. So nanobodies are about 10 times smaller than traditional antibodies, and they bind to their targets with really high affinity, which makes them great for clean immunoprecipitation, super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Proteintech known as Proteintech Genomics. And Proteintech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. So, as a company that is driven by scientists, we are here to support our researchers and our scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. I also want to highlight that Proteintech was awarded the BIOS Rapid Star Award last year for the fastest growing supplier of cytokines, which shows our dedication to the initiative of providing high quality and a large selection of proteins. We also know the importance of product validation, and we offer open access validation data for our customers. When we are testing new antibodies, we do validate them with relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever possible. Also, as antibody and protein experts, we have in depth knowledge of immunoassay applications and cell culture and can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform that we use to manufacture our UniRab recombinant antibodies. With 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world so that you can get started on your experiments right away. So, as a company, we really measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left shows that over our company history, our products are featuring in more and more publications each year. And as of now, we have over 300,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. So with that, I would like to go ahead and introduce our speaker for today, Sreethu Sankar. So Sreethu is a cell biologist by training and previously worked as an industry scientist for 7 years. He has more than a decade of experience in cell culture and cell differentiations and has immense experience in developing stem cell and immune cell differentiation protocols for drug discovery and antibody validations. Sreethu is currently a product manager at Proteintech and manages our humankind growth factors and cytokines portfolio. So with that, I will go ahead and pass it off to you, Sreethu. Thank you so much for being here today and offering us some of your wisdom.
Thank you so much, Afrida. Let me share my screen. Do you see my screen?
Yep, it looks good.
Okay. Hello, everyone. Welcome to this webinar on cell differentiation, the importance of growth factor quality. Uh, I'm Sreethu. I'm a senior product manager at Proteintech. So today, for the next 30 to 40 minutes, which I'm going to talk about, we'll be covering some of the importance of cell differentiation and what is the significance of cell differentiation from an in vitro perspective. And since growth factor being one of the most critical factor required for your cell differentiation, how do we define the quality of these growth factors? And also, one question we often get from customers is to explain the specific activity and why is it important from a growth factor activity perspective, and also how really the expression system or where the source of growth factor really make a difference in your cell differentiation. That's what is going to be covered in this talk for next 30 to 40 minutes. Right. So since we are expecting, our listeners who are starting to do differentiation, some experts in differentiation, maybe just thinking about differentiation, I might also have some very basic slides talking about differentiations in general. So first of all, how do we really define cell differentiation, right? So in nature, cell differentiation is when a group of unspecialized cell gain some functions, and to perform any specialized function, that process of converting an unspecialized cell to a more specialized cell, is that process is called differentiation. And that is specifically influenced in nature through a group of protein that we call as growth factors, and those proteins can influence the different signaling pathways and transcription pathways and everything, so that the cell can perform specific function. For example, if you take an embryonic development, you typically have multiple germ layers, like you have mesoderm, endoderm, ectoderm. From your ectoderm, your neural tissues form. From your mesoderm, maybe cardiac tissues form, and your endoderm, something like pancreas form. So everything functions differently, and it is a different functions in your body. So that process of becoming these specialized cells is differentiation. Um, so what is the real application of studying differentiation or doing this differentiation in vitro? Let's start from the basic itself, right? To understand the process of differentiation is important, because really how an organism developed, not just from a human context, but also from any animal context, how these organisms develop from a single cell to such a complex organisms with multiple organs and tissues that perform various functions. So that process is highly regulated, and how that process happens is important to know. And if you really want to study development, you don't really have access to, a human embryo all the time if you want to say study human development. So what you really want is a scale down model where you can try to learn the same thing from an in vitro system. That's when something like stem cell comes to play, where you can do differentiation in an in vitro condition to all the type of cells that you're interested in, and find out it actually how it develops. It doesn't stop there. Right now, it is also important to study the progression of diseases. Again, if you really want to study some complex diseases, or if you want to study, say, some neurodegenerative diseases for that matter, it is absolutely difficult to get your hands on these valuable samples from a patient. And that too, for a larger population of people, if you want to really understand and study, that's very difficult to get your hands on these samples. So what the potential solution is, use the capacity of certain cells, specifically stem cells, to differentiate into your cell of interest. You can take an adult stem cells, or take a fibroblast cell from a patient who carries a specific mutation, and then you can convert that fibroblast into a pluripotent stem cell by using expression of Yamanaka factors, and then you can use that same cell to further differentiate into your model that you're looking at. So basically, you get your final cell of interest, which carrying the mutation from a patient. And you can use these cells also to understand the disease progression, as well as to screen for drugs. If you're trying to find out what molecules work better for this specific disease, you can use that model system to screen for drugs, rather than depending on patient sample all the time. Um, Another example is like to use it for a very personalized medicine approach. I was recently listening to a talk where when there's cancer for a specific patient, they take a small sample of this cancer, and then they try to develop cancer organoids or spheroids in three-dimensional structures in a, say, 12-well plate or 24-well plate, and try to screen multiple available chemotherapeutic drug in the market, and find out which drug respond to that tumor the best. based on that, they created a dose response curve and used that to dose the patient. And they have found that that is extremely useful to dose the patient with existing drug from a personalized medicine approach. And that's the kind of importance that differentiation have in this market right now. Again, it doesn't really stop there. Now it is moving to actually to the therapeutic application itself, where you can use when there is any problem with some degenerative diseases where your cells become non-functional. The proposed approach to kind of help these kind of patient is to give back cells that is actually functional. And how do we make these cells? Basically to let me start with an example of diabetes. In case of type 1 diabetes, your pancreatic beta cells become non-functional and they won't be able to secrete any insulin. So the idea is to do a cell replacement therapy where you can replace the cells with the non-functional cell with functional cells. And you can generate these functional cells through reprogramming and differentiation of stem cells, and these cells can be given back to the patient. And these are very close to clinical, these approaches. So this is why the differentiation itself in studying differentiation is so critical. Not only just in advanced biology or in disease modeling, all the way up to the treatment of degenerative disease is where it stands right now. Yeah, so we understood the importance of differentiation, but the most and single most critical factor that influence the quality of your differentiation is nothing but growth factors. And how does it really work? Typically, growth factors, like any other proteins, it binds to a receptor on the surface of the cell. And these growth factors functions through signal through your regular PI3K, MAP, or JAK-STAT pathways, and it helps mobilize certain transcription factors to further help in transcribing genes required for differentiation and cell expansion. But it doesn't really, it is not just a function of growth factors, it also influences your cell death pathways, where it can mobilize molecules that can bind to your pro-apoptotic proteins so that it doesn't push the cell to go for apoptosis. Instead, it blocks the apoptosis and induce further expansion and differentiation. Since although we talk about differentiation and the influence of growth factors, I also want to just state that it is also important for growth factors, or in order for the cells to remain in an undifferentiated state, particularly in the case of pluripotent stem cells. If I look at these are like two examples here. One is an iPSC colony. If you really look at a good iPSC colony, what you essentially see is a smooth border with tightly packed cells. The single most important growth factor that influence these nature of iPSCs to remain in this tightly packed colony in an undifferentiated state is BFGF, or fibroblast growth factor. The moment fibroblast growth factors start depleting in your media, you started seeing colonies which showing some spontaneous differentiation, typically into an ectodermal lineage, and a little bit of loosely packed cells. So the idea that what I'm trying to say here, the growth factors not only influences differentiation, it is also critical for making sure that your cells remain in an undifferentiated state, particularly in the case of embryonic stem cells or induced pluripotent stem cells in this case. Now, like all of you understood that it is important, growth factors are, but how do you really define the quality of growth factors when you use it for differentiation? The most important factor to look at in a growth factor is its bioactivity. Typically, how do we define bioactivity is by measuring the ability of these growth factors to induce a biological response in a specific cell group. So what we do, we take a group of cells, so seed it in a 96-well plate. You add growth factors in multiple different concentrations and find out what concentration of a growth factor is required to get the 50% of the maximum response in a cell proliferation assay, or a similar cell proliferation cell death assay. That's what we typically look at. This is an example of a graph that we generate, and from this you can find out, and it is represented by a value called EC50 and effective concentration 50. And lower the EC50 means higher the bioactivity, which means you need only smaller amount of growth factors to induce that good response. So that means your growth factor is more active. But bioactivity is not only the consideration that you have to look at. You need to also look for purity of your growth factors. Typically, almost all growth factors in the market comes with more than 90% purity. 90% is considered good. But what can be the impurities that you might encounter in these growth factors? Typically, if growth factors are made in say in bacterial expression system, which is the most commonly available growth factors, which so in bacterial expression system, the major problem is endotoxins. Bacterial cell wall carries endotoxins, and when you're taking your cells for a clinical experiment or a preclinical experiment where you want to inject it to an animal, endotoxin can generate unwanted immune responses. There might be other host cell proteins. It's a bacterial host cell protein. Although whatever purification method it goes through, there might be remains of these protein. This can actually influence your cell proliferation. Sometimes it leads to apoptosis of cells or unwanted differentiation. The third factor is stability. Stability from a manufacturer point of view, how do we really look at stability? As once you make a protein, you store this protein in different temperatures, and we try to recapitulate how these temperature affect the biological activity of these proteins. So there is some formula that we use, something called Arrhenius equation, where we store for specific temperature at specific time point. You can say that, hey, I do store this as 22-degree room temperature for 3 days or 6 days, which means this can be really extrapolated to a shelf life of protein for say 2 years or 5 years or 10 years. So the bottom line is, the better the stability is really good. So thermally stable growth factors can maintain in your cell culture media for longer durations, which means it can influence your differentiation pathways. If there's something is not as stable as what you want, the problem is you will have to keep giving these product or dosing your cells with growth factors pretty much every day or every 2 days. That's a problem. But it is not just purity, stability, and bioactivity. There is another important factor we need to consider, is the lot consistency of growth factors. The reason is, uh, the growth factors, usually everyone make growth factors in a recombinant way. And in recombinant way, typically when you use a bacterial expression system, there is always chance of introducing animal derived components or undefined components in your manufacturing process. So these undefined components can always influence the consistency of your manufacturing process. What happens when you manufacture the first lot, you get excellent bioactivity. Then you source reagents that is animal derived, say for example, animal serum from a country, and serum is an undefined component, and we have no idea how it's going to influence in a molecular level. Nobody knows how exactly serum influence the manufacturing process. So all these undefined components can have an influence on process of making these proteins. And we need to make sure that, or at least you need to look at suppliers who show you some data in which their multiple lots are tested and found that these lots perform equivalent. Here is an example of how we test for it. We look for bioactivity, and this is a graph of bioactivity, 3 different lots. All 3 lots performed equally good. And this is also the purity gels, which shows that all 3 lots have been exemplary pure. The reason being, we don't really use any undefined component in our manufacturing process. Now let's get a little bit more into differentiation. So when you always start differentiation, this is going to be your expectation. When I started the first differentiation back 8, 9, 10 years back, This was my expectation too, but doesn't happen this way always. What you actually see is, one day it differentiated well, next time it doesn't, next time the cell itself disappeared. I have no idea what happened. But typically in labs, we barely blame it on growth factors. Excuse me. The reason is, um, we look at every other aspect of cells and media and everything, but never really say growth factors might be a problem, but it can actually be a problem. The reason I'll tell you here, typically if you look at most of the differentiation protocol, it asks you to add these growth factors in a specific mass, say 100 nanogram per ml or 50 nanogram per ml of EGF for this specific one, or 20 nanogram per ml of TGF beta 1. The problem is, even if you add the same growth factors from the same supplier in the same exact amount, it doesn't always guarantee that the same amount carry the same amount of active growth factors, because the active units of growth factor can vary between lots, and it depends on different reason, maybe because of the manufacturing process, or maybe because of the inherent variability in the bioassay which measures the bioactivity itself. So the solution is to add growth factors in your media based on specific activity. So what is specific activity and how do we define specific activity? Uh, Specific activity tells you exactly how much growth for active units of growth factors is there in your unit mass that you're taking to add. Say for if you take 100 nanogram per ml of uh, uh, growth factor today, you can exactly measure how many active unit is there in that 100 nanogram of growth factors. So simplest way to calculate this is by using this equation. And this is based on the assumption that 1 unit of growth factor is equivalent to uh, how much growth factor required to generate an EC50 value in 1 ml of your culture. So that is the idea. And often you also see something called IU, or international unit, for specific activity represented in international unit. This is when typically there is international organization which develops standards for growth factors and cytokines. The organizations are WHO and NIBSC. They do extensive validations or testing of these growth factors to find out how much unit is there. So whenever NIBSC sells a sample of growth factors, they say, hey, this is this much unit. So every time that will be consistent. So suppliers or manufacturers like us, we always try to buy these samples and we calibrate it against these standards. So the NIBSC or WHO standard will act as a reference, and we will compare our product with that, and we say, hey, our product in comparison to NIBSC standard, we have these many international unit. So you don't have to really do this calculation, and this can be directly followed for your experiment. But the problem is standards are not available for every growth factors in the market. Many of the this was specifically developed from a therapeutic standpoint, but since we want to be more stringent in our testing, so we make sure that any standard is available in the market, we always try to compare our products with these standards. Now, if you, I want to show you an example here of a simple differentiation of SHSY5I cells into BDNF, into neuronal cells using BDNF and retinoic acid. So clearly, between experiment 1 and experiment 2, there is a big difference. The difference is the amount of differentiation or neurites that is extended, which means the experiment 1 worked, but not as good as experiment 2. What could be the reason? We use the same exact growth factors, same protocol, same set of cells and everything, but still there is a big difference. So the difference here is, the only difference between experiment 1 and 2 was the expression system of BDNF. So the one in experiment 1 where it was not worked very well, which is derived from E. coli, and the other one which you're seeing more differentiation is derived from the human expression system HEC293. And why it is a big difference, I will tell you in my upcoming slides. You'll also see that the BDNF made in E. coli versus a BDNF made in HEC293 cell has six times more bioactivity itself. So why the expression system really matters? Because if you're really working on human stem cell differentiation or any human cell differentiation and you want to use growth factors, so the growth factors are also proteins. In order for it to perform in its best bioactivity, it requires different post-translational modifications. It may be glycosylation, it may be phosphorylation, or it might be just the folding, or whatever it is. But typically, most of the growth factors in the market right now still is available expressed in E. coli. And if you ask me, does it work or not? Yes, it works, but it doesn't work as really the best compared to HEC293. The reason being, in HEC293 cells or in human expression system, it is the machinery to do all these processing. Typically many of the growth factors are secreted as a precursor, and you need a machinery to cleave this precursor properly. Secondly, glycosylation. In bacterial expression system, there is absolutely no glycosylation. Even if you take it to other expression system like mammalian system, the human glycosylation and other cell glycosylation is not similar. There is significant differences. Let me show you a simple example here. One of our most basic growth factor everyone uses is TNF alpha. TNF alpha made in a bacterial cell. What you see here is it's a monomeric form of TNF alpha. However, TNF alpha in its most bioactive form is a 51 kilodalton trimer. So it is first formed as a monomer and then it trimerizes. But the formation of trimer is highly influenced by what expression system that you make it in. If you really look at here, you can actually see some trimers, but most of them remain as monomers. So this trimeric form is so important for its bioactivity. So this is exactly how expression system make a bigger difference to your growth factors. Not just, it doesn't stop there, but if you look at another growth factor like everyone uses, activin A, it's not only just glycosylation. If you look at a very commonly used expression system called CHO, or Chinese hamster ovary cells, which can actually do the glycosylation and some level of post-translational modification, but still in CHO expressed protein, you see more of precursor of activin A. However, in human expression, you see more of activin A dimers and monomers, which are the active form. But you do see that in CHO expressed protein as well, but you end up seeing a lot more of these precursor in CHO expressed protein. And another key example I want to show here is beta cellulin. It's a classic example. It's a very important growth factor required for differentiation of your pluripotent stem cell to the beta cells of pancreas, which I said has been, it's widely been explored as an alternate cell replacement therapy for diabetes. So the example what I'm trying to show here is this a comparison of human beta cellulin expressed in human expression system versus bacterial expression system. Beta cellulin being a super glycosylated protein, when it expressed in bacterial system, you don't see that glycosylated form. This is a typical Western blot of how a glycosylated protein shows up. And is it just about the glycosylation? How does it really impact the protein performance or the functionality? So let me show you some bioactivity data here. We have compared it against two most popular beta cell in the market, which is expressed in E. coli. What we have observed is when it is glycosylated and expressed in human expression system or HEC293 cells, the bioactivity is at least 50 times more compared to one competitor that we looked at. Another is 100 times more compared to another bacterially expressed beta cell in the market. So this is exactly the difference that will make when the protein is expressed, a human protein is expressed in a human expression system versus a bacterial expression system. And how does it actually translate? There is another example here of macrophage generation from human monocytes. Monocytes can be converted to macrophage in the presence of growth factor IL-34. So what we did is we took the monocyte from human PBMCs and tried to make differentiated. And basically to look at bioactivity itself, it was 8 times more active compared to something that is made in E. coli. And if you look at the data speaks for itself. There you see a lot more, at least 10 times more of macrophages compared to the macrophages generated using a bacterially derived IL-34. So this is the clear difference. What it actually signifies is you can even use some lower amount of growth factors to get better differentiation when you source it from an expression system that is more human with more right post-translational modifications and folding. So that can impact your cost, that can impact your work, that can impact your timeline. uh, I also want to show another example of IL-2, which is very critical for expansion of T cells. And this is the data generated by one of our customers. And they found that when the IL-2 is made in human expression system, it is 40 times more bioactive, and it is able to generate or expand T cell much more significantly than something that is made in E. coli. This is probably my last example, but I want to talk a little bit more about BMP2. This is an independent, a group of researchers trying to compare all the available BMP2 in the market and find out the bioactivity and functionality of BMP2. What they found is when BMP2 is also a highly glycosylated protein, and almost out of every BMP2 in the market, the HEC293 derived or humankind BMP2 was the most bioactive in the market. But doesn't just stop there. And there are also other researchers looked at what is the significance of these glycosylation on differentiation. Let's look at in vitro osteoblast differentiation. If you look at the left side, this image, it is quite clear that what they did, this site is actually the glycosylation site of BMP2, and compared to wild type BMP2, and when you mutate this site, this clearly influences the total differentiation of osteoblast. And in mutated glycosylation mutated form, the differentiation was in excellent compared to this. And they also try to give BMP2 separately, and they found that it is again gaining back that function. So it clearly shows that the influence of these post-translational modification is much significant than we ever thought on differentiation pathways. But it's going one more step ahead, not from in vitro to in vivo applications, where one of our customer looked at, they took these BMP2 and they injected it to mouse to understand bone regeneration using BMP2, and they found that the glycosylation and non-glycosylated BMP2 behaves very differently when it comes to bone regeneration. And they looked at bone calcification and how much size the bones are forming, and they clearly found that always the glycosylated BMP2 performed better than the non-glycosylated BMP2. So this is basically what that how the difference it is actually making when we really look at expression system before deciding on starting your differentiation. So always make sure that you try to find out if it's a glycosylated or something that is required post-translational modification. Try to source it from expression system that is more human. So like these problems that what I told here, we are trying to solve these problems through the product line that we are making here in Proteintech, which is humankind. How humankind is different is every humankind protein that we have, we derive everything from HEC293 cells. All of them are human proteins. It has much better bioactivity and stability. Every data that I've shown you there is humankind data. We have proven advantages in stem cell differentiation, and it is already cited over 1,000 times. And not only that, all humankind proteins are animal component free. Like I said, the lot to lot consistency is such a critical factor for your differentiation. We make sure that we don't add any undefined components in our manufacturing process. So it is totally animal component free. Xeno free means there is no non-human components added at all. There is no bacterially derived components or recombinant components in a manufacturing process. It is totally endotoxin free because since we don't use bacterial expression system, there is no endotoxin at all. And also, we don't use affinity tags. Typically, in order to purify protein easily, everyone use affinity tag. We also think that this tag itself can and sometimes interfere with the bioactivity of the protein. Although the process is a bit complex, we decided that we will go without the use of any tags for our making our proteins. So I just want to conclude by showing you some examples of some specific humankind protein that will be really helpful. This is a thermostable FGF2, which Proteintech have a patent on this. The reason for making a thermostable version of FGF2 is FGF2 typically degrades in the media quite quickly, like I think it's 7 hours or something is it half-life. So as I said, the moment FGF2 degrades in the media, your cell will start spontaneous differentiation. In order to avoid these, it is important for FGF2 to remain in the media for longer duration. So we developed this FGF2 by doing some mutations, like around 8 or 9 mutations in the backbone of FGF2, and we found that this FGF2 is at least 12 times more active than the international standard itself, and it is. We also looked at its ability to maintain iPSCs, and how long it can remain in the media in compared to normal BFGF. We found that when the normal BFGF completely degrades in the media in a day, where the humankind thermostable FGF remains in the media for at least 3 to 4 days. So if you're really doing a lot of stem cell differentiation, this can have a big impact on your the whole process of differentiation, as well as it can save significant amount of money, because you don't have to add FGF2 as often as you would otherwise with the normal FGF. Also, it has induced higher expansion of embryonic stem cells. This is the customer generated data. They have clearly seen that the humankind FGF2 was able to expand embryonic stem cells in a better manner. Also, since I talk about purity, some proteins are notorious to be known to get it pure. So for example, Win3A being one of them, which is very critical for several differentiation pathways. And we looked at all the Win3As available in the market. Everything was like 75, 70 or 85% pure. So we really wanted to make the purest form of Win3A in the market. And although with a lot of difficulties, our team came up with a plan to make the purest Win3A available in the market right now. And we compared it against several Win3As in the market, even something that claims close to 80% purity. We looked at in the gel, and we found that on the right side here, it is so impure. It is even way impure than actually what they claim. And compared to humankind, if you really look at humankind Win3A here, it is basically no impurities in the gel. So it is actually one of the most purest Win3A available in the market. So these are just two highlights product I wanted to show you all, and how we are really making an effort to help our customers change their differentiations. So in summary, what I'm trying to say here is ensure growth factors are sourced from human expression system, particularly when you're working with human cells. If you're working on human embryonic stem cells or iPSCs or MSCs, make sure that you source these growth factors from a place where there is, which is expressed in HEC293 cells or any other human cells. Opt for animal component-free cytokines. So animal-derived components can definitely influence differentiation if there is any components coming from the serum that is being used in manufacturing. So it's always better to go for animal component-free product. Whenever possible, or I wouldn't say whenever possible, there is, so right now, since we are giving the equation too, always try to look for the units or international units of your cytokines or growth factors to ensure that you have more consistent results during your repeated experiments. So follow the equation that I've showed you, or looked at the manufacturer and find out if they already give something in international unit. Also, I just want to make a comment here. Sometimes manufacturers gives IU or international unit as a blanket value, but it is not true. International unit, although it is measured, it should be a lot specific value. So whenever you source any growth factors, always look at the CFA or the certificate of analysis, and find that every lot is tested against it, and find you need to make sure that not just for one product, one IU value exists. It is not like that. Every lot will have smaller differences in their international values as well. So in the bottom line, higher the bioactivity, purity, and stability is better for your differentiation. So with that, I am stopping here. I will hand it over to Afrida.
Great. Thank you so much, Sreethu, for that wonderful talk. I think this is such an important topic to cover. I remember, when I was in the lab, our lab sometimes got really stuck in our ways, and we didn't consider, changing up reagents really when we were troubleshooting or trying something that might work better. So, thank you so much for highlighting that for us today. I think we can go ahead and begin the Q&A session. There is quite a few questions, so hopefully, we get to most of them. If we do not get to your question live, I apologize. But at the very end, I will be providing Srithu's email, so you can always email him directly for any kind of follow-up questions that you guys might have. The first question that I wanted to start with was there were a couple questions regarding how we calculated the EC50. So, what are we measuring and how do we calculate that?
Sorry, can you repeat that question again?
Yeah, so how are we calculating EC50?
How are we calculating? So typically what we do is like we develop something called a bioassay. Bioassay is where you seed cells, a specific set of cells that is actually we think that will respond to the growth factor, and then you grow that cell, and then you add different concentrations of growth factors, and you observe how that concentrations influence the growth of the cell, and then you create a standard curve, and then you calculate how much growth factor was required to really get 50% of the maximum response that cell have shown. So if the cell has grown from 10 to 100, and how much growth factor was required to grow to 50 cells, that was a whole concept of, but the process is much longer, and it involves multiple steps of bioassay validations and testings, and so it's a very complex process to explain in a simple word, but yeah,
Yeah,
that's a
no, thank you. No, you explained that quite well. Um, awesome. So our next question is, could individual genetic differences between healthy humans affect stem cell differentiation? And if so, do you have any thoughts on how you could account for that?
the individual genetic differences can affect differentiations. Yes, because I have seen, even in some publications, I have seen not just, if you look at a cell which is derived from a male patient versus a female patient, that itself respond differently. So I don't know if there is a simple ways to address that issue, unless you just have to include multiple controls to make sure or eliminate that difference. That's all I can say now, but that genetic difference is not something that we can control. Yeah.
Yeah. Um, so I guess a follow-up to your response to the EC50, there was a question saying, how is this different from IC50?
So EC50 is effective concentration. IC50 is it's, I think it is for the inhibitory concentration. It is typically measured for how much is required for inhibiting. That's what I remember. So okay, but I mean, if that's specific question, I can try to it. But EC50 is looking for the effect of something, but it is slightly different. Typically, IC50 values are look for drug which induces us or stops a specific pathways.
Great. Okay, thank you. Um, our next question is there any concern that HEC293 cells are immortalized cancer cells and thus not really the same as normal/control human cells?
So HEC293 cells are not, uh, it's not a cancer cell, it's a human embryonic kidney cell. But then the concern is no specific concern, because what we are essentially trying to explore by expressing these proteins in HEC293 is to uh, get the human machinery which can really glycosylate and fold and express these protein. That is the part that we are trying to take from that cell. So it's not of a big concern because we have also done a lot of further analysis on these proteins that we express through these HEC293 system. Even there are therapeutic proteins available in the market which is made in HEC293 system. So it's not a big worry for me.
Great, thanks. So there's a question about our catalog. So in the Proteintech catalog, can I find all cytokines that are normally produced in E. coli system that are also available in the humankind version?
So you usually, if you're, I wouldn't claim that every thing will have it, but we are trying to expand our catalog. So you will find most of the critical growth factors required for differentiation in our portfolio. But we are actually expanding the portfolio quite quickly.
Great, thank you. There's also a question about our FGF beta. Do you know the stability or how stable it is at 4 degrees Celsius?
Are you asking about FGF basic?
Basic, yes, sorry, I wrote it
So
down wrong.
uh, I don't really remember on top of my mind how long it is stable in 4 degree, but I'm sure it should be like at least a month stable in 4 degree. That's what I remember.
Okay,
But the
great,
stability
thank you.
varies between proteins. I mean, I should be able to send that information to that person if they can write to me.
Okay, sounds good. Um, So there's a question about regarding bioactivity. So in the assay, the cells have to be do they have to be FBS deprived serum or completely depleted or just with low level?
So it depends on bio. So typically, it's always ideal not to have any other, factors that influence cell growth so that you can clearly measure the effect of your growth factors. Because if you look at FBS will itself have a lot of growth factors in it. So it's better to have serum free or serum deprived cells for your bioassay.
Great, thank you. Um, there's one question that's asking what factors contribute to choosing the source of your stem cells?
What factors contribute to choosing the source of the stem cell? Uh, so I don't know what exactly meant by source of the stem cell, because
I'm
if you
assuming like where the stem cells are originate.
so I think it all depends on what experiment that you're trying to perform. Because I think it's a very broad question to answer. But if you're uh, looking at, say, stem cell, just for the stem cell differentiation, some episomal iPSCs available typically differentiate very well. Sometimes, if you're looking for, or maybe you can think about where that iPS, say for example, if I'm talking specifically at iPSCs, in from what cell that iPSC is generated, is that cell basically uh, was it a mesodermal origin or an endodermal origin? So you can look at that kind of which germ layer origin that original cell was in. That kind of gives you an idea of where the differentiation would be going. So it's a bit of a complex question to answer. I understand that because again, if you're looking for a therapeutic application, sometimes from an autologous stem cell therapy point, you might just take the patient cell and give it back to the patient. From an allogenic point, you take the cell from a patient and you try to make it for a larger population. So it varies quite a lot, depends on your end experiment.
Great. Um, there was one question that was asking, what would be the effect of using, instead of living cells, immortalized cells to study, for example, how how they grow in presence of a stimuli or co-cultured with other living cells?
Can you repeat that question again?
Yeah, so instead of using living cells, immortalized cells to study, for example, how the growth in presence of stimuli or co-cultured with other living cells. So instead of using like primary cells, utilizing immortalized cells.
How the effect? Well, I think there'll definitely be effect. So we have looked at a lot of our products using cell lines, which is immortalized, of course. Some of the cell lines which are immortalized. But as far as you include your controls, you can actually clearly make that differences. show the differences, like how it is impacting, the molecules are impacting. But I strongly believe that as far as these cells express these receptors and the back growth factors are able to bind those receptors, can actually influence the growth of immortalized cells too. And I mean, even cancer cell, when people try to make cancer spheroids, which are definitely immortalized cells, or which has uncontrollable division, people use combinations of various growth factors to study the proliferation or make organoids or cancer organoids. So it definitely influences, but there will be definitely a difference influence, but you're talking two things in a very different context, you know studying a cancer versus. So yeah, that's what it is.
Great, thank you. There's a specific question centered around mouse macrophages, so primary mouse macrophages, saying, I work with primary mouse macrophages, and I would like to know if you're supposed to take out the growth factors after differentiation. In my experience, the macrophages detach and die once I take it out, even after differentiation.
Oh, that's a very specific question.
Yeah, I
uh,
didn't know if you had like a general, like in general, for most cells, are you supposed
So
to take
typically,
out?
we don't take out growth factors, because once a cell tend to use growth factors, it gets deprive itself of growth factors, and it degrades itself. So we don't really remove growth factors in many of the differentiation. Um, yeah, and the end of differentiation, it depends on your application. After maybe like a 50-day differentiation, what is your final application? You probably want to uh, co-culture it with something else, but we still don't really take out growth factors in any of our experiment that at least we have done in-house or in my past that I have worked on.
Great. And then kind of a follow-up question on adding the growth factors into media. Is it better to create a complete media with growth factors and keep using that, or add single-use frozen aliquots of them?
I would always recommend single-use frozen aliquots, because unless the we don't nobody really study the stability of growth factors for a longer period in the media. But usually, if you are just using your media for maybe a week or so, it might be still stable. But again, it depends on growth factors. There are certain growth factors which are extremely unstable. Like if you take, as I said, Win3F, for example, it doesn't stay for longer time. Some TGF betas, for example, it doesn't remain in the media for longer time. So it require other additional factors, which is usually present in the reconstitution buffer that we make to stabilize these proteins. So it is very growth factor dependent, but in a general sense, I would always recommend to have frozen aliquots and add it to your differentiation media rather than keeping it in the complete media.
Great, thank you. Um, Another media question. So, when do you recommend filtering the culture medium? Before or after adding supplements and growth factors? Is there anything else to consider?
So I would say filter it before uh, adding the growth factors. And if you're adding serum, I imagine you might filtering your serum before. So, or else otherwise it is not really. So most of the readily available media is pre-filtered. Even the growth factors that we sell is also pre-filtered and tested for sterility and everything. So yeah, I know some labs follow different protocols when they add all the supplements and filter, but that's not actually required. The only other difference would be when you are making media from something like a media powder, where you expect there will be some what is so some suspended particles in your media that might you want to remove. That case, you can just make the media and then filter it and then add the growth factors. But typically, you don't have to filter it after adding supplements.
Okay.
Yeah.
I think we have time for maybe a couple more questions. So the next question is, where can you find, you were talking about those standards. So where can you find these standards that were developed by HUNIPC?
So NIBSC and WHO. So there's a website for NIBSE. So that's where you can find these standards. So that's when from where we are also buying the standard. So I think MHRA is the one in the UK is one who validate these standards, and they this I think WHO works closely with them too. get hands on these standards, and they sell it too. So NIBSC is the place where you can find these standards.
Great. Okay.
Yeah.
And then the last question is um, asking a question about one of your slides. So on slide 22, you mentioned by some mutation in FGF, you achieved a longer half-life in this molecule. How about its activity? It didn't cause any unwanted occurrence?
No, so yeah, I think we have tested multiple mutants and everything, and one of the mutant was even better bioactive than the international standard itself. And we didn't face any problem with activity, but that wouldn't be the case for every cytokine, I would imagine. So you might want to develop multiple mutants and test and find out. It may not work for everything, but FGF basic being something that degrades in the media so quickly, I think we really wanted to solve that problem. That's why we came up with this idea of generating a thermostable version by mutating the backbone.
Awesome. All right. Well, thank you so much. I think those are all the questions that we have time for. There are just a couple more slides that we need to go over. But as I mentioned on the very last slide, I will have Srithu's email. So please do feel free to email him at any point with any of the questions that we did not get to live, or if you guys have any other additional questions for him. So with the last few minutes that we have left, I just want to share some more information and resources that are available to you guys. So if you are interested in learning more about cell culture and specific differentiation protocols, please do check out our video hub on our website. There you will find on-demand webinars that you can peruse through. So for example, we have had past webinars with external speakers discussing oligodendrocyte differentiation, culturing iPSCs, culturing tumor organoids, culturing brain organoids, and so much more. So definitely check that resource out. And then as an attendee of today's webinar, you will receive 2 follow-up emails. The first follow-up email will be a certificate of attendance, and then the second follow-up email will include a recording of this webinar, as well as a link where you can request a free sample of 1 humankind protein from a selected list, and this would be 2 micrograms of that protein. Or if you're very eager to request your sample right away, you can scan this QR code and do that today if you would like to. But we do ask, please allow 1 week for the follow-up emails to be sent out. So with that, I would just like to thank everyone for attending today's webinar. Thank you so much, Sreethu, for taking the time for this training. It was really, really informative, and I learned quite a bit, and I'm hoping our audience did as well. If you do have any further questions about the event itself, please feel free to email us at events@ptglab.com. If you have any humankind related or cell differentiation questions, feel free to email Sreethu at sreethu@ptglab.com. And if you liked today's webinar and want to learn about any upcoming events and webinars that we have, please do follow us on social media because we post about all our upcoming events on there. So with that, I'll go ahead and end the webinar. I hope everyone has a great rest of your day or evening, wherever you're tuning in from the world. Take care.